everyone. Welcome to today's Variety Hour show for June. We're really excited about all of the speakers and projects we've got coming up today. So today's Variety Hour, we've got a variety of talks from across the world, but they seem to be have fallen into big categories of marine, freshwater, AI, and bioacoustics, passive acoustic stuff. So should be an interesting mix of talks today. We're going to start off in Australia with Andrew, and who's going to be talking about the work he's been leading in Kakadu National Park. I'm really excited about this because that's where my conservation tech journey started as well. So I really can't wait to hear what you guys have been doing. So Andrew, yeah, I'll hand it over to you.
Thanks, Stephanie, and thanks for inviting me. I'm actually so excited to be here. Seems like a really awesome community that was Dan Morris actually kindly introduced me to. So I'll just
Dan has been sending a lot of excellent speakers our way. So yeah, thanks, Dan. Big shout out.
Yeah,
if anyone, and also general shout out to anyone. If you know of like a cool project, feel free to ping it to us so that we can feature it as well. All right, over to you.
Thank you. So my name's Andrew. I'm from the Australian Government Department of Climate Change, Energy, Environment and Water. And all the work I'm talking about today was done by the supervising scientist, which is a branch within that department. So yeah, I'm going to be talking about how we're using drones, underwater cameras, AI, to monitor freshwater fish assemblages up in Kakadu National Park. And I've sort of kept it at a high level, so I'll drop my contact details in the chat after if anyone wants to dive a little bit deeper. But yeah, just to let everyone know, if you're not familiar where it is, so Kakadu National Park, it's World Heritage listed. It's got highly significant cultural and ecological values. It's in the north of Australia, so where that pin is, which is the wet dry tropics. So if we zoom a little bit closer in, the area or all the sites that we're working in to monitor fish are around this floodplain system here, the Magilla floodplain. So wet and dry seasons, you've got October to sort of March, April of like intense rainfall, monsoonal, where everything fills up, floodplain fills up, connects all the water bodies and streams and creek systems. You get this large migration of fish and resources into the system during the wet, and everything's moving around. And during the dry season, as things recede, these water bodies become isolated and they act as refuges for these fish assemblages. So yeah, I'm going to be talking about the fish monitoring program that is done, and you might have seen the map. A lot of it surrounds the Ranger uranium mine that's based in that's surrounded by Kakadu National Park. So their sites up and downstream of the mine. So just to give you a bit of a background, the two different water bodies I'm going to be talking about today are channel billabongs, which are the deeper, more narrow billabongs that are permanent throughout the whole year, and shallow lowland billabongs, which are shallower, full of aquatic vegetation, and they only connect to the wider system during periods of high flow when the stream will actually backflow into it. So there's been sort of some traditional monitoring practices over the past 20, 30 years, where in channel billabongs, they would actually have somebody lay in a boat and count fish through that little dome that you can see there. And in the lowland billabongs, they would actually put these nets out and push these pop nets in and trigger them a day later, pull out all the fish, weigh them, and all the aquatic vegetation as well, so you can relate that habitat variable to the fish. But for obvious reasons, this big crocodile in the middle, it's no longer safe to do that. So about 5 years ago, they transitioned to a new method of measuring the fish assemblage and the aquatic vegetation. So following what's pretty established in marine ecosystems is using underwater cameras in these freshwater tropical billabongs. So putting a GoPro on a stand, deploying it in benthic and surface locations to get different fish assemblages within the water column. And then, just moving over to that method, subsequently capturing, 300 plus hours of video a year. That was sort of my job when I first started to sit in front of a computer screen, identify all the fish species that you account and count how many there were. So, moving on from that and saying, this is going to become a backlog of, human time and also skill set in the taxonomy and being able to identify fish. How can we, automate that process and try and get a machine to do it? And that's where we really kicked off our project, which we've called WILDLABS.NET, which is, trying to get AI to identify fish and give me some time back in the day. So I'm sure most people are familiar with, computer vision. Everyone's heard about ChatGPT. But I guess the point of this slide is to say that, you need a training data set to train a neural network to identify what you want it to learn. And when we started this project, there were no publicly available data sets of tropical freshwater fish from Kakadu. So we had to start from scratch. And that process is, taking an image from one of our videos, identifying the fish, annotating and labeling it to pass through that model, then use that model to identify new video and save me time. So there's lots of different ways you can annotate and label images. The least expensive and time-consuming one is image classification, where you just tag the whole image. You could then put a box around the image for object detection, a box around each fish in the image, sorry. Or, the more time-intensive one of actually putting polygons around all the fish or objects in the image. So each with their pros and cons, and we sort of explored both the object detection and instant segmentation approach of using boxes and polygons. But for today, I'm going to focus on sort of where we moved to with instant segmentation. So when we started actually looking at what our videos look like, the bounding boxes, and we have a data set that's publicly available that I'll put a link to in the chat. When the fish are, sort of sitting around individually, the bounding boxes work really well. But when you start to get this complex imagery where they're all overlapping each other, the bounding boxes overlap and, it tends to fall over. So that's where we needed the polygons to segment out each fish if we wanted to get, high performance in actually counting each of those individual fish. Some of the, key things that we learned along the way is, when you start generating that training data set, there was some key steps that we sort of figured out that I thought might be useful to share with people. Is, there's that data preparation stage where you got hundreds of hours of videos and you got to extract frames from it. So, coming up with a way to extract all those images and be able to track them for each year and billabong and location, and carry that metadata along. We came up with a set of labeling guidelines for fish because there wasn't, a stock standard one. And this is all in the GitHub repo that I'll share too. What actually makes a good label for a machine learning model? So, key features, the orientation of the fish, how deep it is in the image. And then also baking in the taxonomy that, a human would use. So, I can identify a fish to species in an image. Baking that sort of information into the labeling guidelines to say, okay, well, I wouldn't be able to do it to species level, so we'll only do it to genus, and just creating that set of criteria. So, we've generated this training data set. We've, labeled all this data, and then we used Microsoft's Azure Machine Learning Studio to sort of handle all the machine learning parts, so the ecologists can focus on the ecology, and we let the data scientists focus on the data science. But essentially, we've come to this place where we have, a workflow that we can take that model, we get new videos, to say our 300 hours of videos from the field, and we can put it into a storage account and then spin up multiple virtual machines in the cloud to, get 500,000 frames analyzed by a model, write it all back to a database, and then, do our analysis, which is, really nice place to be in. And then this was meant to be a video, so it's just an image. This is, it's sort of just an example of the instant segmentation model, putting polygons around fish and, automating what one of us would do. So moving on from fish, I mentioned, aquatic vegetation. So, you're putting a camera out, and it's really important to know what the habitat is of that fish assemblage that you're characterizing, for one, to understand, any changes in fish assemblages related to water quality, or are they related to changes in habitat? So, a traditional approach with those nets would be to, get this slither along the edge of the bank of the billabong, and, get replicates and get a sample, a representative sample of the site. But we moved to using drones with multispectral sensors. So now, I think most people are familiar. You can go to the shop and buy a drone from relatively cheap, or you can go and buy these multispectral sensors that are a little bit different. They got, like this one in the picture here has got 6 lenses that have different wavelengths in them. So what does that look like? So the latest one we're using is an agro wing sensor with on a Sony A7R camera, and what you get is you get this picture that's in the middle, and then post-processing you can actually do different band ratios to take those 6 images and get 14 bands of the one image across the blue, green, red edge and near infrared spectrums. So you can stack all those 14 images on top of each other, and you've got all these 14 different bands that are useful for extracting different things from the image. We also collect high resolution RGB imagery. So if when we actually flew it and looked at it, you're sort of getting this high level of detail than you would from standard satellite imagery, where if you zoom in, you can actually see flowers, individual lily pads, the submerged vegetation, emergent vegetation, which was really cool to see. But I guess, the analysis to begin with started with the multispectral imagery. So if we've got this image on the left, we take the near infrared, one of the red bands and the green band, and look at that image there. It's expressing all those floating lilies really well. And then if you take the near infrared, the red edge and the green with that image on the right, it's expressing the submerged vegetation really well. So you can use those different combinations of the bands to see the different vegetation types. And then along with a range of indices that you could also calculate as well, and you can use that to spectral threshold on the whole mosaic and get a classified map for the whole site, which is to pixel level, which is a lot more data than we were getting on the ground. And I will just note that these are broad categories, not at the species level, but work towards those species level classifications. And then I guess the next step is when I actually saw that, that's essentially just like a picture with its training data. They're just labels for an image. So we thought, okay, let's just chop it up, throw it through a neural network, and then train a model, and then just pass new imagery through it. So starting to build an AI model to process that drone imagery as well. So we can, then you can start sampling in temporal and spatial space a lot better. And then, I guess just to show it side by side, you can see there's, vastly improved scales of vegetation mapping, and you can relate it to each of the individual cameras, or, the whole site, or whatever you want to look at. And sorry, I know I've probably gone a little bit over, but probably no time for questions.
Slightly over, but it was we respect it when it's a really interesting project, and you guys are doing such, like, there are so many different aspects of the technology layers that you're exploring. Are you, my question is kind of a bit random, but are you a government employee?
Yeah.
So I'm fascinated that that part of your work is like pushing the boundaries so far with the different technologies. However, well, I feel like I should ask you at least one question. What I would love to know is, like, what are you most excited about the next steps of these projects, of how you're applying technology, whether it's the drone remote sensing stuff, or with the fish AI work?
I guess with everything I've presented today, that was sort of where we first entered into, and what I haven't shown is where we actually have gone on to. So we've so that was all single camera stuff. So we've actually just run a little bit of a sprint project to get a stereo camera design, and then get the AI to hit both the stereo cameras and then get biomass estimates, which is cool to automate that, because that's twice the amount of data, and a whole new metric of measuring abundance with the cameras that we're not getting with the single stuff. And then I guess being able to pick up, so that AI model I was showing, it's actually been applied in the base model weights for the fish has been applied in so many different areas, from identifying imported fish in bags at the border to in aquaculture farms for fish, counting, moving between ponds. You can just pick it up and you can use it anywhere. It doesn't need to be fresh water. So I think that's the most exciting thing, is just seeing, once you invest a bit of time to get these models up and running, they can be applied anywhere, any domain. That's any other other part of the world, related to fish or vegetation or anything.
Amazing. Thank you so much for your talk and sharing. And are you on Wild Labs? Are you okay with people popping? We'll start a thread, but if people have questions, you're okay with people popping you messages?
Yeah. Sounds good.
Amazing. Thanks, Andrew. Okay, we're going on to our next speaker, which is Adrien, talking to us about nocturnal bird migrations and combination of citizen science, AI, radar, integrating a lot of different pieces of technology and people here. Adrien, do you want to take it away?
Yes, thank you. Thank you to the WILDLABS community and to Stephanie to let us present NBM for Nocturnal Bird Migration. The project is to automate the tracking of birds by night with and for ornithologists in Europe. The with and for is very important, you will see it. So first, a bit of an introduction. Maybe you know it, but nocturnal migration, it's a quite crazy phenomenon occurring twice a year throughout the world. It concerns billions of birds. And as you can see on the right images, some radar just get 3 million birds predicted moving during the night of the 16th of May in the US. And on the left, you can see the different corridors at fall in Europe. So nocturnal migration is a crazy phenomenon that you can see when the weather is very bad. As you can see on this picture, this is on an island, the Seal Island, where the birds stop because of the weather, and you can see thousands of them with different species. This is quite the only way to see the nocturnal migration, because using your binoculars is quite impossible, apart if you have thermal binoculars or you are during a very bright moon with a bright city close by. It's quite impossible to see the birds moving. And to follow migration, you have to stay awake all night long to try to identify the call of the birds, but it's quite challenging. You have to stay all night long awoken during a period of 2 or 3 months and try to identify what's happening. So we have one issue, how to follow and identify, birds while they migrate by night. There is a solution. This is bioacoustic. You have a recorder, you put a micro, and you put over your balcony, in your garden, over your roof, and you listen to the sound of the night. The sound of the night will be on the band and an audio file that you can transform into a spectrogram and see the little shape you can see on the right, such as the call of this common ring plover. This is very nice, and particularly during the pandemic, because birds could not go outside. So they put a lot of micro and recorders out, and they had a lot of information to explore and to go through. It is really exciting. However, it is very time-consuming and challenging. Indeed, for a night of 8 or 10 hours, you will have 30 minutes at least of analysis to see what birds have moved through your garden or your balcony. And you have to be quite expert in identifying birds, because you have no context, you have no shape, you have nothing to identify the birds apart from this shape and the sound. So we would like to help ornithologists to go through this topic, nocturnal migration, thanks to the new technologies and particularly AI. To do so, the NBM project was born, and we had some goals. So the goals were to give access to nocturnal migration tracking to everyone, and to generate a great amount of data on this unknown phenomenon. Indeed, as you have understood, it is occurring by night, so we don't really know about this. We know that species are now moving by night because they avoid some trees. We don't know how many, we don't know when, so this is to generate data in order to make scientific analysis, but also to track the birds on large spatial and temporal scale. Indeed, with micro, you can go everywhere, anywhere, without putting a human in danger. And so the NGO was born thanks to and based on 3 communities. The first community is a community of birders, the ornithologists. The second community is a community of 10 data scientists. And the last community, making the link between birders and data scientists is the developers. How they work together? First, the birders. There is the Discord group chat. If you want to join the project, I will put it at the end. So first, the ornithologists, they have an important job. It's to label sounds in order to train a model. We have a data set now of more than 10,000 labels for more than 90 species, among which you can find the common crane or the bittern that are passing through your balcony in Europe. This labeling was done with the help of Audacity, and there is a documentation how you can help us in order to have more and more data, because as presented in the present presentation and as presented every time here in the variety hours, data is the most difficult and important things to have. Then the developers create a platform. The volunteers, of course, everyone is volunteer here. There are no salary or paid person. The volunteer developers developed a platform in order to put the data with the annotation at a place where it could be easy to use. This is an open source platform, so you can find the code on the left, and you can participate in putting your data on. All the data are accessible, so if you want to train a model to automate the following of a migration in your country, you can use the data that have been recording with the volunteer builders. And finally, and most importantly, with the data, we develop an AI model based on the data set and I would say the skill we had. So we had this slide made by Luis, who is not present. So the principle is the following. You have your recording, as I told you, in the morning, you get your SD card, you put it on your computer, and the script will transform your recording into a spectrogram. This is the visual representation of the sound. After putting this into a spectrogram, we can analyze it as an image. So thanks to a faster CNN algorithm, we are now able to detect and classify birds, such as this black-bellied plover, with quite a high confidence. And we are very proud of it because we have a very nice detection and classification precision, and it is now available and freely available for everyone. And that was our main goal: help birders to go birding. That was kind of the most important thing to us. So these achievements is now done, but we have, of course, things to continue. First, we want to continue to label data because we are only, I told you, 90 species, and there are a lot more moving through, and the least common one are very difficult to catch. So we have to train our model. And this is the second goal: improve the AI by continuously train the AI, refactor the code with the new technologies, because as you may know, with the different presentation, the technologies are moving very fastly. And we want to facilitate the access to the AI, and we are helped by the M33 organization in France to go not without Kaggle, but without another solution in order to help the birders going through the technology. And then we want to analyze our model, because this is important to know what are exactly the precision of the detection, what is the classification rate. So we will do statistics on it. And finally, you have understood that this is a collaborative work. We are working with a lot of birders throughout France and Europe, and we continue this collaboration with a French national project, with the Natural History Museum, the Bird Protection Link, and Biophonia, and are part of the Nocturnal Migration Tracking Working Group. So if one of these three goals interests you, feel free to contact the team in order to join this fantastic project. Thank you for listening. Here is the founding team. If you have any question, feel free to ask them. Here are the links if you want.
Sorry, I jumped in too soon. Sorry, I got excited.
No. This was kind of the hand and just.
So Louis has been dropping heaps of links as you've been talking, so it's a great tag team effort, and Carly, he's picking up questions Carly's been asking. My question before we move on, because I'm conscious of time, this is not your day job, is it? Or you're not a bird conservationist by background,
Yeah.
are you? How did you?
This is not our day job, yeah.
So how did you, as someone who comes from like more of the tech side, right? How did you find your way into this?
Oh, that's a nice question. In all the funding team, our job is closely related to the topic. The people from Biophonia work in Bioacoustic. Luis is a PhD candidate, and Hervé and I are project manager, teacher, working and ornithologist. But this is the patient who brought us there. We are all birders, and we want to help other birders with our skills. So we have a day job and a night job. The night job is NBM.
Awesome. Okay, I'm going to pop you back into the chat. If people have questions for Adrien and Louis, please do ask them in the chat. Thank you so much. That was really interesting. But now I'm going to throw over to Ellie, who has thematically aligned her quiz today with our themes of bioacoustics, right?
I have, that's right. We're going to have a sound quiz. But first, we're going to have a little ad break. Here we go, everybody. It's time for a Wild Labs ad break. We have the voice of Wild Labs, Jake Burton, here with us today. Jake, are you ready to tell us about what's going on with Wild Labs?
Yes, thanks, Ellie. Hi, everyone. So the first ad break is come work with us at Wild Labs. We're seeking an experienced project manager to join us at Fauna and Flora in delivering projects under our cross-sector funding and events programs. So if you have the skills to bring together a cross-sector community and help us provide critical resources to support collaboration and innovation in the global conservation technology community, apply by the 9th of July for consideration. And you can find all the details on how to apply at Wild Labs. Great, and next it's almost time for our favorite time of the year, which is the annual Tech for Wildlife Photo Challenge. And that's coming this July. For the past 7 years, you've shared your projects, stories, and successes with us. And this year, we're making our photo challenge bigger and better than ever to help you showcase your conservation tech work to the world. Stay tuned on Wild Labs for details on fresh updates coming to the challenge this year, and as we continue to introduce new submission categories, awards, and exciting opportunities for finalists. And get ready to tell us how you are using Tech for Wildlife. Back to you, Ellie.
All right, thank you very much, Jake. That's right. All of those, the links for our job opportunity is on Wild Labs right now in our resources section, along with many other opportunities that you'll want to explore. And keep your eyes on Wild Labs for more information about the photo challenge that will be coming very soon. And you'll also be meeting our new intern very soon, Maddie, who will be helping us roll out the photo challenge this year. But for now, it's time for a game. The sound is working, right? Everybody heard the air horn? Great. The sound has never been more important because this week we're playing bioacoustics. We're going to guess who is making these sounds that have been sent to us, courtesy of Carly, who is here at every Wild Labs event. She's here right now. She was a big help in putting this together. Carly, would you like to say anything about how you contributed to this with Rainforest Connection?
Hello, yeah, literally coming to you live from appointment after appointment because I'm so committed. So yeah, I have put together a collection of different sounds with accompanying spectrograms. A bunch of them have come from our Artimon platform, where we have lots of public templates that people can use for training their own species detection models, with some help from other open source sound libraries from around the world. So hope you enjoy.
All right, thank you, Carly. And with that, we will get to our first bioacoustics. Now, everybody, I want to tell you, we're looking for real specific answers in the chat, okay? I don't want to hear like, oh, that's a big cat. Oh, that's a bird. I need you to guess specific types of animals. I need species, okay? Don't put anything general in the chat or we'll be very disappointed in you. Here we go.
Orca?
What do we?
Lauren's saying orca? A door from Louis?
A good guess, a good guess. Anything else?
You are very kind, Ellie. Whale from Adrien, beluga from Lauren is getting very specific with her whale calls.
It is. It's a beluga. Someone got it.
Lauren is sitting right on top.
All right,
Yes,
Lauren, you're
Lauren's
in the lead
queen.
right now. Here we go. Here comes the next one. I'll play it again. That was a short one.
Lion? No. Tiger? Cow? Cow from Lauren? Kate says lion. Consolata says cow. Steve says hippo. Alistair, Indian guar. I don't know how to say that. Alistair, you might have to jump in and pronounce that for me. Jake is saying hamster. Paul, deer. Warthog from Esther.
Okay, no one's getting it. You have 3 more seconds.
Wait, can you give us a hint?
Right.
What continent would you find it on?
You would find these in, I believe, South America. I hope that's
Bear
right.
from Anna. Sea lion from Dan. And Alistair does not know how to say that species either, so I'm in the clip. Humor.
It is. It's a jaguar.
Big. Okay, we had a lot of big cat suggestions. Okay.
So those were close, but nobody quite got credit.
Oh no, I'm sorry, Steve got it. Steve,
congratulations, Steve.
Sorry,
you got
Steve.
It.
Did Steve get in time, everyone?
Yes.
Yes. Okay, Steve.
Good job. Okay, so Lauren and Steve in the lead right now. Okay, here we go.
Titty monkey from Tremaine. Sea lion from Apex Primate. I wonder who that is. Any other? I thought it was a bird of some description. Egret.
I'll play it one more time while people guess. It's playing in the background. Please keep guessing.
Monkey from Kate. Lauren says parrot. Tropical bird from Adrian. Louis says egret. Toucan from Stefan.
Longtime attendees of Variety Hour are going to be so mad when they find out what this is.
Is it? Because it's Carly. Is it? Is it? I've just forgotten what Carly.
It is. This one I had to put one in specifically for Carly. This is the black and white ruffed lemur. That is them. I believe that is them doing their famous roar shriek that Carly talked about. I'm not 100% sure. Carly, if you would like to confirm if that was a roar shriek, feel free.
It's actually the quack, not the roar squeak.
I think, okay. So it looks like Carly is literally in the doctor's office. So we'll get confirmation from her in the chat.
It's the quack. It's the quack.
That is the quack, folks. A new sound from the lemurs, the quack sound.
It's a mating call. It's what happens when the females reject the males.
Oh, okay. That's a fun little detail. Okay, next up, who do we have? Who's this, folks?
McCall from Adrian?
A what?
Sounds like Carly's dentist appointment. Trumpeter.
I'll play it one more time in the back. Please keep guessing, everyone.
I'm with Jake as well. Is it a lyrebird? No? Okay, crow from Consolata, parrot from Kate, trumpeter from Tremaine, heron from Dan,
I mean, we've got people in the right genre
Goose.
of animals.
Southeast Asia is the hint. Southeast Asia, guys. Oh,
I can't see its name because the Zoom
black
window is
hornbill.
blocking it. The black hornbill. Everybody, I don't know where they live. Are they in Southeast Asia?
I think we have, next time we do this, we have categories of specific species and type of animal, right?
Look, that everyone's just complaining now because
No, it's
they're
great.
getting it wrong. Everybody needs to strengthen their listening skills, okay?
Okay,
Everybody,
we'll get better.
We're starting it out with a real challenge because I believe in you guys and I think you should be challenged. Okay, I think this is the last one. So everybody be on your best behavior and try to do a good job.
Ellie, that's the wrong spectrogram.
Oh, it is?
Look, we like to be specific. Okay, right.
All right, well, nobody look at the spectrogram. Just listen to the beautiful sound.
So, that is wonderful. No idea of this. Very old car from Adrien. Is it a mammal? Flight of the Bumblebees from Alistair?
Oh, that's
Insect from Anna?
it's close. I'll bring up the picture in a second.
Bees from Francesca?
It is an insect.
Bees
I'll
from
give you
Consolata?
that.
Mosquito. No, it can't be mosquito. No.
It's
Fly?
it's a type of fly. It's the Mediterranean fruit fly.
All
That's
I
what
can
they
say.
sound like. I don't know what to tell you. That's good. Everyone, oh, you're telling me everyone doesn't know the sound of the Mediterranean fruit fly.
We are,
you know,
but
hearing it one time.
I think this is my favorite quiz in like quite some time. Carly, thank you for your help. Sorry, you haven't done your final.
Yes. Oh, hang on. My Zoom thing, I keep moving it so I can see the actual slides. Yes, thank you again to Carly and the Rainforest Connection for letting us play with your data. And now, back to the show.
Sorry, before we move on, can you explain that last image?
Oh, yes. Every time I've been including an image from Raccoon Saloon, which is a YouTube channel of an abandoned house where there are a lot of raccoons that are eating food, and there are lots of babies on there. So that's a plug for Raccoon Saloon. I've been waiting for someone to ask me what those pictures are from every time.
Look, we know this, that other people might not know our team's fondness for raccoons and possums, but okay, right, we've got to move on. Thank you. That was glorious. Carly, thank you so much. Good work, Ellie. We're going to Steve now for some talk about what you guys are working on with building community around oceans and passive acoustic monitoring, right?
Yeah, thanks, Stephanie. So I'm going to share my screen and I will need to make sure that the sound is working as well. Excellent. Right. Okay. So this is a report from the first ever World Oceans Passive Acoustic Monitoring Day. And I'm giving it on behalf of both myself, but also Miles Parsons from the Australian Institute of Marine Science, whose birthday it is. And I believe it's also probably after midnight. So he's looking forward. Now, I've just got to just give you a very brief background to WAPAM Day. So first of all, this came about partly through about a 15-year community development project called the International Quiet Ocean Experiment, where bioacousticians working in the oceans have come together, first of all, to imagine what the oceans would sound like in the absence of human activity, but then through a whole series of working groups to try to develop standards, develop approaches, develop ways of monitoring biodiversity hotspots, ways of monitoring sound in the Arctic, and so on. And so this time last year, I was getting ready to go off to COP, which was in November, December last year, for a project with Google Arts and Culture called Calling in Our Corals. where the idea here was that we were going to try and take COP27 by storm with the underwater acoustics of Sharm el-Sheikh, the Red Sea, where the COP event was taking place. Now, this is a citizen science platform that allows anyone on the planet to listen to recordings of coral reefs from a whole range of different places around the world, and to interact with them, to score the sounds, to try to help us to build an understanding of patterns in biodiversity. For example, looking at how marine protected areas change biodiversity with reducing fishing impacts. So in launching Calling Our Corals, we found that we could actually live stream the sound of the Red Sea to the COP arena. We're very lucky to find Zayed Gohari, who had live streamed the Red Hot Chili Peppers from the pyramids and had all the gear to live stream, which then allowed us to be able to have this live feed from the reefs of the Red Sea into the Blue Zone arena. So this was the shell that we'd built that allowed anyone to be able to come and swim in the sounds of the Red Sea. And this turned out to be a really effective way of connecting people who may not have really spent time in marine environments. One of the most exciting collaborations that came from it was with Inawa Hunikui, who's the president of the Federation of the Hunikui Peoples in the Amazon, who's currently introducing electric boats to the Amazon. And so experimentally, we're able to look at whether the switch to electric engines is having a boost to the catches in the fisheries because of the reduction in noise pollution. So that's where I was this time last year, while Miles was developing the Global Library of Underwater Biological Sounds, GLUBS, which is a collaboration of, at the moment, 17 different scientists who manage GLUBS, but it involves now over 40 institutes collaborating together to try to build this global underwater library. And so over the last 20 years or so, we're aware that there are many more people listening and recording from the world's oceans. And so by collating recordings, we're trying to then explore across all taxa what those soniferous species might be. And there's currently 36,078 species being considered, some of which we know produce sound. Orca, beluga whales, brilliant examples. Some of which we suspect might make sounds. So there are 24,000 species of fish that we don't have recordings of, but we suspect they probably can make sound based on the phylogeny of those species. But also then marine invertebrates and a whole range of other taxa. The idea here is that we can submit this to the World Register of Marine Species to bring acoustics into our understanding of all of these species. So Miles and I both went to a coral reef symposium last summer, and together over a couple of beers came up with the idea of WAPAM Day. We wanted to try and find ways of bringing this growing knowledge of marine acoustics to the world's attention, and collecting and connecting the bioacousticians that were working on this. So this year, on the 8th of June, we were able to hit World Ocean Day with a collective effort to record. So we ended up with 300 acousticians, supported by 150 PIs, 100 institutes, 34 countries, taking recordings around the world, all on that single day in history. So there's a very brief film that explains what we were doing, which I'll play now. Hopefully you can hear this. Hi, I'm Steve Simpson, and I'm one of the co-founders of WAPAM Day, where we're getting people to listen to the ocean all around the world on World Ocean Day.
Hi, I'm Filipa Samaha from the University of Iceland. I'm listening to whales in the subarctic to assess habitat use.
I'm listening to and playing back underwater sounds help build Australian coral reef resilience.
I'm listening to the individually distinctive signature whistles of dolphins to better understand their communication system.
We are deploying hydrophones to listen to the soundscapes of coral reefs to try to discriminate between different types and qualities of habitat.
we are deploying and recovering underwater recording instruments to understand how do mesophotic reefs sound and how do they differ from shallow water reefs.
I'm listening to humpback whale song in Southern Africa.
We're listening to understand fish communities in Hawaii.
We're listening to soundscapes of the Canadian Arctic under the sea ice, to listen to sounds made by marine fauna, as well as to measure underwater noise from ships once the sea ice breaks up.
And today we are recording sound in freshwater pond to study the ecology, amphibians, fishes, and invertebrates.
Hi, I'm Miles Parsons from the Australian Institute of Marine Science in Perth and co-founder of Wapanda. We can't wait to collate these recordings taken on the same day all around the world to help build a new global understanding of noise pollution, biodiversity and ocean health.
So that's where we go next. The question is, what will we hear? And this is something that we would love anyone here who's interested to join. The next stage is that we're going to be collating these recordings, putting them into a public repository, and then having some online workshops where artists, scientists, physicists, machine learning specialists can collaborate to try to pick apart the new species that we might hear. So we'll definitely be doing a lot of guessing the species. And ultimately hoping to be able to report back on what World Ocean Day sounded like in 2023. Next year, we're planning to go bigger and better. And one of the ideas that we're ultimately building towards is whether we could ever have a World Ocean Quiet Day, inspired by the Nyepi New Year's Day in Bali, where all motorized vehicles remain quiet for a day. So that's the direction of travel. Be really keen to have any of you involved in this next step of analyzing, and particularly those interested in machine learning and using AI to analyze very large data sets that we'll have. Okay. Thank you.
Amazing, Steve. My only comment is that your team is very good at naming things. Like your names are so satisfying to say, so my team appreciates it very much. But that was also just such a powerful video, and I wonder if there's some crossovers with our Tech for Wildlife photo challenge, because we get a lot of entries. So I wonder if we could build some bridges or help. Yeah, there's a collaboration there, I feel. So we'll follow up with you afterwards. But if anyone has any questions for Steve, but also, Steve, we'd love to hear updates as you progress as well and sharing with our community. So please keep in touch with us to hear more. But we've got one final speaker, Francesca, who's been talking about her work on WildLabs, which is why we asked her along to share what she's been working on and like the challenges that you're having in your work with penguins and Acoustic.
Yeah.
So do you want to take it away?
Definitely. So hello to everyone. Thank you, Steph, for inviting me in this really nice talk. And I want to say that I really like the quiz of Alice. So I'm Francesca Terranova, and I'm studying bioacoustics and penguins in my PhD project that is about passive acoustic monitoring as a tool for support Spanish forms conservation. Just to clarify, because maybe Spanish-forms is not a word that everyone knows, but Spanish-forms are penguins, so aquatic flightless birds that everyone knows, I think. And it's important to know that 10 out of these 18 species of penguins nowadays are classified as vulnerable or endangered. And between these 10 species, there is the African penguins, that is the target species of my PhD project. And that is a monogamous bird that inhabits the coast of South Africa and Namibia. And as you can see from this graph, this is the reason why now it's really endangered. There was a big decline of this species in the 1960s, between 1960 and 1980. And for this reason, South African government is pressing a lot for creating and biodiversity management plan for this species. And this is the reason why I'm doing my project, just to understand how to monitor these species without disturbing them. And for this reason, I'm using passive acoustic monitoring. So basically, I use the autonomous record unit that I placed around the colony. And the pro are that you could use the chip devices and you could record continuously also during bad weather and during night especially. The colony in which I did my data collection was a Stony Point colony. It is a mainland African penguin colony. It is composed by around 1,700 breeding pairs. And my objectives are to use passive acoustic monitoring to estimate the number of breeding individuals and the presence of chicks inside the colony. In order to do this, I did my data collection during between February and May in 2023. And as you can see, this is the Stony Point Nature Reserve. That is a beautiful place, but as you can see, is a coastal side. And I placed 8 different song meter micro, and I recorded between 6 p.m. and 7 a.m. basically, because from previous studies we know that the vocal peak is around sunset and sunrise hour, and I did 30 minutes long recordings. Now I'm full of recordings, of course, and my proposal is to use a particular song that is in the vocal repertoire of African penguins. This is called exotic display song. And in the period of data collection, I also collect data about the nesting activity. So if the nests are occupied or not, and the presence or absence of chicks and egg. Just to show to you, this is an exotic display song emitted by an African penguin. I hope you hear that. And more in particular, this is the composition of the song. So basically, this is a song that is used for territoriality and also for the attraction of mates. Is mainly uttered by males, but is still we still don't know exactly. And as you can see, it's composed by different syllables. So basically, we have the short syllable A, then the long syllable B, the syllable C, that is an inspiration syllable. Oh, there is another one. So basically, after the data collection, we use a monitor package to automated detection of ADS that did work pretty well, because basically we use the syllable B as template. The challenge is that there is a big variability between the ADS uttered between individuals, but also the same individuals could change the composition of the song, emitting 2 B syllables, for example, or also just emitting A syllables, as you can see from the spectrograms. So there is an ADS that is correctly detected by monitor, and then an ADS with just the syllable A. And another big challenge of this day was collected just detect the detection of the recordings with wind. That in my case is a big problem because I can just erase the first killers of the spectrograms because the ideas are low frequency sounds. And at the same time, I want to say thank you to all the people that helped me in WILDLABS.NET and also in Acoustic Exchange, because they give to me a lot of insights that now I'm trying to detect the detection of the wind, because basically I have to detect the wind and just remove from my dataset, since there is a false positive detection that confuse, of course, the final analysis. That's all from my side. This is the beginning of my result about my PhD, so we will see. And thank you. And if you wanna just for the question, there is the email or the Twitter account. Thank you.
Amazing. Thank you so much, Francesca, for sharing like what you're working on and some of the challenges. And it's great that you've been finding help through WILDLABS and the Bioacoustic Stack Exchange in your project. I'm gonna give you the same question I asked Andrew at the start. What are you most excited about that's coming up in your work?
In the next field season, I think we will do always passive acoustic monitoring in the same colony, but with also the just focus on the density of the nest and how connect the density of the nest with the passive acoustic, with the vocal activity basically of the African penguins. So yeah, we will see, because
oh, sorry, keep going. I interrupted.
Depending also from the results of this first field season, we will just maybe focus in some area or not, because basically what we want to do is that all the stuff that we are doing on this Stony Point Nature Reserve just translate in all the remote places that in which there are the African penguin colonies, because basically a lot of African penguin colonies are in the remote islands that are not so accessible. So the protocol of passive acoustic monitoring will help to the monitoring of these species in the remote places.
Amazing. As you progress, please keep us updated and tell us how your work's going and how the sort of challenges that you're facing and overcoming and lessons you're learning along the way. It would be great to keep in touch as you keep going. All right. Look at us almost being on time for once. Well done, everyone. To wrap us up, I just want to say a big thank you to all our speakers, particularly Andrew, who joined at 1 a.m. Australian time. So big kudos to that level of commitment. And we will see you all here next month in July. It's around the 20th. I can't quite remember the date, but all of the dates are on WILDLABS if you just search Variety Hour. And as always, suggestions for speakers and projects or requests are very, very welcome. But we'll see you next month. Thanks, everyone.